So we left off on Monday talking about transcription and translation. And so we said with the initiation of translation, what you're looking at is an mRNA molecule. It comes together with the small and large subunits of the ribosome, and now we can begin translation. So when translation is in process, what happens is a tRNA, the function of the tRNA is to bring an amino acid to the ribosome. The tRNA brings the amino acid to the ribosome and it lands in the A site. Okay. There's an expenditure of energy, and the amino acid in the P site is transferred to the A site, because remember, when you start to build things, you have to expend energy. And so then we have an empty tRNA in the P site. The A site contains the polypeptide. We spend more energy so that we can move the mRNA along. And now what happens is the empty tRNA is in the E site, or the exit site, so it exits the ribosome. The P site now contains the growing polypeptide, and we would have room for a new tRNA to come in. So you'd continue to have this growing polypeptide. So really, every time you want to add an amino acid, just at the ribosome, you have to expend at least 2 GTP. So it costs a lot of energy to make proteins. When we get to a stop codon, so when a stop codon is what enters the A site, what happens is we don't bring in a tRNA, because there are no tRNAs that match the stop codon. Sometimes the stop codon is also called nonsense, because it doesn't encode for anything. So there are proteins that will recognize the fact that no tRNA is brought in, that no tRNA will match. They are called release factors, and they remove the polypeptide from the tRNA. When that happens, that allows everything to dissociate. It allows the whole ribosome complex to fall apart. And so then a new molecule of mRNA can come in and cause the ribosome to reform. So we do reuse the ribosome after making a single protein. So we don't just make a whole new ribosome again. Okay. In prokaryotic systems, one of the differences between bacteria and archaea and eukaryotes is the fact that we can do transcription and translation at the same time. Right? It makes bacteria and archaea much more efficient and faster because they can do that at the same time. What we can also have is multiple ribosomes that form on the same mRNA. So you can make multiple copies of the protein from one molecule of mRNA. And that we call that a polysome. Multiple ribosomes on a single molecule of mRNA. And so what they're showing is it would look something like this. At the beginning of the mRNA, you have a ribosome. Towards the end, you have a ribosome. You have some ribosomes in the middle. Of course, near the end, we have a long polypeptide. At the beginning, we have a short polypeptide. But we can make multiple copies of the protein at the same time. So not only can we have transcription and translation occurring at the same time, we can be doing translation with multiple ribosomes at the same time. Okay, and we also talked about ribosomal RNA as being important for structure of the ribosome. It's a complex of ribosomal RNA and proteins. More specifically, the rRNA can have individual roles in the ribosome depending on what kind of rRNA it is. So the 16S rRNA, this is unique in prokaryotes. Eukaryotic systems do not have 16S rRNA except for in their mitochondria or chloroplasts. Right? It's another one of those lines of evidence that supports endosymbiosis. In the nucleus and the major sites of protein synthesis in eukaryotic cells, we use an 18S RNA. In bacteria and archaea, we use a 16S rRNA, which is, again, has to do with the size. It helps to facilitate initiation by holding the mRNA in place. So remember, the ribosome itself, there's 2 subunits, a small and a large. Each of those subunits is made of multiple proteins and multiple pieces of rRNA. So the 16S of rRNA helps to hold the mRNA in place on the ribosome so that the tRNA can come into the right place. Ribosomal RNA can also be involved in positioning the tRNAs to help guide them to the A, P, and E sites. And finally, one thing that RNAs can do that a lot of people don't think of, so not only do RNAs carry the message, we can translate RNA into proteins. tRNA, right, serves as a transfer molecule. It's a transporter that can bring us amino acids. Ribosomal RNA is structural to set up the ribosome, but many types of RNA also have enzymatic function. They can catalyze reactions. Normally, we think of enzymes as being proteins, a protein that catalyzes a reaction. But in the case of forming proteins, RNAs actually have the catalytic activity. So an RNA molecule can act as an enzyme. So it catalyzes peptide bond formation. They also interact with the other proteins called elongation factors that assist with protein synthesis. So ribosomal RNA, for the test, of course, all we had talked about previously was that it helps to form the ribosome. So that's all I'm looking for. But ribosomal RNA actually has far more functions than just helping to form the ribosome. Okay. Finally, what we have to think about, this little last step when we're building a protein, we talked about that you have a primary structure that is just the chain of amino acids. You have a secondary structure where you might have alpha helices or beta sheets. But eventually, in order for a protein to function, it has to have a shape. And we know in biology that if you change the shape of a protein, you change its function. And by changing its function, you could make it no longer work at all. Okay? And sometimes just a single amino acid change can change the shape of a protein so it no longer functions. You've heard of sickle cell anemia. That's the classic disease we talk about. It's a single nucleotide mutation that leads to a single amino acid mutation that causes your red blood cells to go from being nice and round and fluffy to being curved and not able to carry oxygen. So protein shape is critically important. The example that a lot of people probably see on a daily basis, or maybe at least a weekly basis, if you cook eggs, the reason an egg goes from being runny to being hard is because you have denatured the proteins. You've changed their shape. And when you change their shape, again, you change their function. And a lot of times when you change the shape, and you've cooked an egg, you can't uncook an egg. You take a cold egg, you can heat it up all you want, nice and cooked, ooh, hard-boiled egg, delicious. Put it in the refrigerator, it doesn't go back to being a liquid. Right? It's a solid. You can't change it. And so when things happen inside of your cell that start to change the shape of your proteins, let's say you start to get too hot, and the shape of your proteins starts to change. Okay? You have to be able to fix that, stop it from happening. And as your proteins are being made, you have to make sure they get folded into the appropriate shape, because shape is so tightly linked to function. So in order to do that, we have a class of proteins called chaperones. What chaperones do is they ensure that proteins fold appropriately as they are being generated, or right after they are generated. Chaperones can also fix partially denatured proteins. Like I used the example of if it starts to get hot and your proteins start to denature because of heat, there are certain chaperones that are turned on when it's hot. We express them at high levels when it's hot so that they can help refold proteins that have started to misfold. And this is part of a stress response that lots of cells have to tolerate heat. So chaperones are really, really important proteins that get us our final three-dimensional structure. And what happens if a chaperone messes up? Well, if it messes up at this point, when we're first trying to make the protein, if the chaperone doesn't fold it properly, usually the cell will destroy that protein. It will chop it up and reuse the amino acids. But if it gets to a point where the chaperones can't keep up with a stressful situation, like we've started to cook our E. coli, the cell will eventually die. So that's why, even though cells like E. coli have a really great stress response, as long as you cook your food properly, you will kill the bacteria because you'll denature their proteins, you'll cause their cytoplasm to boil, you'll cause a cell wall to burst. It's a lovely process, but they can't recover from it. But if you start to warm them up just a little bit, they can tolerate it for a while because of these proteins. And so here they're showing you an improperly folded protein. You have some other proteins that can be activated and move the improperly folded protein to the chaperone, which can properly fold the protein. So again, chaperones and protein folding are critically important for functioning of a cell. Okay. So what I'd like you to do now is take just a minute, because I told you